Guys, it is really cold in here. Yeah, man, it's really cold in here.
Alright, so those clips will make more sense as I go through these slides. So in class we learned that sex isn't determined by our chromosomes, like whether we have XX or XY, but by the genes on them, and in some cases the environment too. So if you're a reptile, fish, or amphibian, your sex might depend on the temperature of your incubation. This is called temperature-dependent sex determination, or TSD. So our textbook describes 3 general patterns of TSD. If we're looking at case 1, which is exhibited in some turtles, FT on the bottom is the female determining temperature. This means at lower temperatures, a majority of the turtles will be female. MT is the male determining temperature, which means that at higher temperatures, most of the turtles will be male. TP in the middle is the pivotal temperature. Here, the population is going to be 50-50 males and females. And there's not always a single pivotal temperature, like in case 3, we have 2. The pivotal temperature is the focus of most TSD studies because it's crucial in sex determination. Knowing the incubation and pivotal temperatures, you can predict the sex distribution of a population. There's actually a study on a certain lizard species that found no pivotal temperature. A majority of the population was always female. So while this seems simple, the study proposes that this might be a little more complicated than we thought. So how does this actually work? In these species, the sex is determined by the production of sex hormones during embryo development, specifically in the thermosensitive period. Aromatase is an enzyme that converts androgens, which are male sex steroids like testosterone, to estrogens, which are female sex steroids like estradiol, during the thermosensitive period. Temperature variations can increase aromatase activity. So if an individual has lower aromatase, less of their male sex steroids will get converted to female, and they'll be a male. If an individual has higher aromatase, then more of their male sex steroids will be converted to female, and they'll be a female. There are other factors that affect aromatase activity, but temperature has the greatest effect. So TSD was first described in 1966, but it was seen as an imperfection in sex determination, so it was mostly ignored until the 1980s. Now, we're trying to better understand what affects TSD, because we know that there are environmental implications for these reptile, fish, and amphibian populations. One study suggests that TSD actually maximizes reproductive success in some lizards, so things that mess with it can be potentially dangerous to those populations. Environmental pollutants that replace sex steroids can change the sex determination, and climate change can also be an issue, as temperatures rise and populations get closer and closer to higher majorities of either males or females. So saying all that, there's still a lot we don't know about TSD, but I hope that you learned something today. Thank you, and here are my citations.